Vice President of Research here, and she's going to discuss the recent in vivo data for GT-02287 as a best-in-class small molecule for the treatment of GBA Parkinson's disease and other neurodegenerative diseases. So Joanne, please take it away.
Thank you very much, Hartaj, and thanks very much to our previous two speakers who have undoubtedly made my job a lot easier. So as Hartaj said, I'm going to introduce you to our GBA modulator, GT-02287, which is currently in the clinic as a treatment for GBA1 Parkinson's disease. So this molecule was actually discovered from our proprietary computational drug discovery platform. And this is actually all of our molecules which were in our pipeline were discovered employing this platform. So we're really delighted that GT-02287 is now in the clinic as a proof of concept of the utility of this platform. And before I really get going, I'd really like to acknowledge the generous support of various foundations that have financially contributed to this work over the years. And they include the Michael J. Fox Foundation, the Silverstein Foundation, Inner Swiss, and Eurostars. So I don't think I need to go through this slide at all, given the two excellent previous presentations. Just to reiterate that we think that restoring GCase function is expected to stop or slow the progression of Parkinson's disease, and we're hoping that that's what our candidate molecule GT-02287 is going to be able to do. So we believe that GT-02287 addresses the primary pathophysiological effects in the disease cascade resulting from GCase function that we've heard quite a lot about already from our previous two speakers. And in a plethora of both cell-based experiments, some of them using cells derived from patients, and also animal models in which we have modeled GBA1 Parkinson's disease, we have seen that our compound can ameliorate lots of the effects of GCase dysfunction. So, for example, we've been able to see effects on endoplasmic reticulum stress, which is caused when misfolded GCase is not able to traffic through the cell to the lysosome where it needs to be to perform its function. This causes an accumulation of toxic lipids, the substrate of the enzyme, as well as alpha-synuclein accumulation. And we've also been able to show in many, many experiments that we can prevent this from happening with our compound. The lysosomal dysfunction that ensues from all of this build-up of alpha-synuclein and lipids and GCase not being where it's supposed to be, we can also stop this happening with our compound. And we can also stop the accumulation of mitochondrial reactive oxygen species. It's become clear recently that not only is GCase important in the lysosome, but it needs to get to the mitochondria as well, where it's important in energy homeostasis. So having misfunctional GCase seems to affect mitochondria, which we know are very, very important in neurodegenerative disease, including Parkinson's disease. And then in animal models, we've been able to show that our compound affects neuroinflammation, that it prevents neuronal cell death, particularly dopaminergic neuronal cell death, and it prevents the ensuing motor deficits from all this cascade of pathology. So because of this, we expect GT-02287 to have potential as a disease-modifying therapy in Parkinson's disease, as well as other neurodegenerative diseases where a deficit in GCase function has been implicated. So we think that our compound is has a best-in-class profile of the GCase targeting drug candidates that we know about to date. And it's an orally available, it's a brain penetrant small molecule, and it's actually an allosteric GCase regulator. It binds away from the active site, so it doesn't interfere with substrate processing, and by a mechanism that we heard about earlier, this compound actually chaperones GKs to the lysosome where it needs to be to perform its function. And as I already mentioned, this compound's already in phase 1 clinical trials. So I'd like to present to you some data in a model which we've used a lot to test this compound, and also follow-on compounds. And it's a model where we've tried to model GBA1 Parkinson's disease in normal animals. So the way we've done this is we've used a compound called CBE, which is an irreversible inhibitor of the GCase enzyme. So we administer this compound to the animals, and this takes out some of the activity of the GCase enzyme, and it's irreversible. So we can forget about that GCase in the model. We've taken it out of function, and then we are testing our compound on the effect on the remaining GCase, which hasn't been inhibited by this compound. So we are trying to model the reduction in GCase activity that we see in GBA1 Parkinson's disease. And what we see in this model, and what we see with our compound, is everything that you'd like to see really in a model of GBA1 Parkinson's disease. And with our compound, we see everything that you'd like to see with a molecule which is treating this disease. So for example, we see an increase in glucosylceramide, which is a toxic lipid substrate, and we're able to prevent this with our molecule. We've heard a lot about aggregated alpha-synuclein in this meeting, and we, in our model, we see a big increase in aggregated alpha-synuclein, and we're able to prevent this with our molecule. Likewise, we see a decrease in the number of dopaminergic neurons. We see this with tyrosine hydroxylase staining. This is tyrosine hydroxylase is an enzyme important in the metabolism or the making of dopamine, and we're able to prevent the loss of these dopaminergic neurons. Maybe unsurprisingly, because we're losing dopaminergic neurons, we also lose striatal dopamine, which we can prevent with our molecule. We see an increase in neuroinflammation. We see this with EBA1 staining. It's a marker of microgliosis. Again, we prevent it with our molecule. We're also preventing loss of neurons in the cortex. Cortex is, of course, also very important in Parkinson's disease, and we as we've heard, we see cognitive decline, especially in GBA1 Parkinson's disease. So it's important that we can rescue these neurons in the cortex as well. Very exciting. In our model, we see an increase in neurofilament light chain in the plasma. So neurofilament light chain is emerging as a marker of neurodegeneration, and the fact that we can see it in the plasma suggests that this could be a biomarker for the effect of our compound in an accessible body fluid, i.e. the plasma. Obviously, the brain is not accessible, and there are some issues with taking cerebral spinal fluid as well. And then just to round this off, as you might expect, given the hypothesis of how this all works and given all the other effects that we've seen, we are able to rescue the motor deficits that we see in this model. So when we measure the amount of time a mouse can hang on to a wire or an upturned cage lid, when we administer the CBE, the latency to fall, that's the red bar, is very much reduced, and then we bring this back up towards control levels with our compound. And then in a beam walk experiment, which is measuring coordination, so we count the number of foot slips that the animals make, they're much less coordinated in this model, and we are able to prevent this with our compound. Now, all of the experiments the experiment that I just described to you was actually done by giving our compound at the same time when we introduced the toxic insult. So the really important thing to know is does our molecule work when the disease process is already underway? So we wanted to take a look at this. And first of all, we took a look at this in cultured dopamine neurons. So we're looking at the effect of our compound in cultured neurons in a dish. And we've cultured these neurons. And then, as I've just described to you, in the animal model, we've added CBE to these neurons, again, to knock down some of the GCase activity. And then in addition, in this model, we've added alpha-synuclein preformed fibrils. So as we heard from Dr. Otero, that when the synuclein aggregates together and this becomes more kind of toxic and pathological, we've added this into the culture as well. And we can see, comparing the black control bars and the gray bars, that when we add CBE and alpha-synuclein, we get a reduction in neuronal survival, we get a reduction in the neurite network, and we also get an increase in lysosomal pathology. Now, when we add our compound after the start of the experiment, so when all the disease process is in place, on the next day, essentially, we are still able to rescue these effects on neuronal survival network and lysosomal pathology. What's very interesting, given the discussion that we've just had about idiopathic versus GBA Parkinson's disease, we actually see very, very similar results when we treat these neurons not with CBE. So we're not really affecting the GCase, but when we just add alpha-synuclein preformed fibrils, and we see very similar effects in the cell-based model, and we see very similar effects of our compound, i.e. we can rescue this effect and prevent these neurons from dying even in a delayed treatment paradigm. So we've now taken this into animals, this therapeutic paradigm, and again, we've given the animals CBE, but this time quite a low amount of CBE, half the dose that we gave in the previous in vivo experiment I described to you, and we're only giving it every other day. And as we have done with the cell culture experiments, we've also administered alpha-synuclein, preformed fibrils, and we've injected these straight into the brain, again to mimic what happens in Parkinson's disease, and specifically in this experiment, GBA1 Parkinson's disease. And then once we've got this whole disease cascade going, we started to treat these animals with our compound after either a 4-day delay or an 8-day delay. And then we measured the motor function of these animals in a wire hang test, which I described to you before. So we tested the animals at either day 14 of the experiment, which is about a week after we started treating with compound, and at day 27 of the experiments, which is at about 3 weeks after we started treating with the compound. And then again, we used this plasma, we did the motor performance, the wire hang, and then we also measured plasma NFL as a measure of neurodegeneration. So what we saw when we tested these animals in the wire hang test on day 14, so this is a week after we started treating with the compound, we saw in the red bars here that the latency to fall is really short. The animals really can't hang onto a wire at all when we administer CBE and alpha-synuclein together. This is additive compared with when we just look at alpha-synuclein alone in the light gray bars, or CBE alone in the dark gray bars. So we're adding to this model where we've got CBE and alpha-synuclein together, we're starting to treat the animals with compound either 4 days or 8 days after we first start the experiment. And you can see that both after 4 days in the paradigm where the treatment started at 4 days, or when the treatment started at 8 days, and we measure the performance a week later, we can see that in both paradigms, we managed to more or less rescue completely the motor deficit in these animals. The animals where treatment started at 4 days are doing a little bit better than the animals where treatment started at 8 days. When we measure the performance of these animals 2 weeks later, so 3 weeks after we started, approximately after we started treating with compound, again, we're comparing the red bar with the blue bar. We see that these animals that have been treated in this therapeutic paradigm have still, still very, very good motor function. And what's also interesting, if we compare the dark green bar at day 27 and the dark green bar at day 14, these animals seem to be doing a little bit better. So these are the same animals that we tested at day 14 and day 27. So it seems that as we continue to treat, that the animals do even better. And then again, we measured the plasma neurofilament light chain levels at sacrifice at the end of the experiment. And we were very pleased to see again that the plasma neurofilament light chain levels pretty much mirrored what we saw in the motor test. So when we have low latency to fall, we have high levels of neurofilament light chain. And then when the latency to fall went back to control levels, so did the levels of plasma neurofilament light chain. So as I mentioned, this compound is already in the clinic. It's in a phase 1 trial in healthy volunteers. The upcoming milestones for this trial are this quarter. We expect to complete the single ascending dose part of the phase 1 trial and to start the multiple ascending dose part of this trial. And in the coming quarter, we expect to finish the MAD part of the trial, and we also expect to be able to collect some data looking for effects on GCase activity and levels, even in these healthy volunteers, because well, I may not have mentioned, but I should have mentioned that our compound seems to act on non-mutated GCase in terms of helping it get to the lysosome and increasing activity as it does in mutated GCase. So we want to look at GCase activity and levels in healthy volunteers as a first in human proof of concept. And then in the third quarter of this year, spanning out, or the second half of this year, spanning out to the first half of next year, we want to add a patient cohort onto the phase 1 trial, where we will start to look at biomarkers that you can measure then in patients. So, for example, neurofilament light chain, given what we've seen in our preclinical models, and other markers of, say, for example, neuroinflammation and so on, and how these lipid substrates are moving as well. And then later on, we expect to have a US IND filing, and then to start our phase 1B / phase 2 clinical trial. So I don't have time for the conclusions, actually, but I think we can go to the question and answer session. But just before I finish, I'd like to point out that obviously I didn't do all this work, and it's been carried out by past and present members of the Gain team over the years. And I would particularly like to call out Dr. Beatriz Guzman, who has produced all of the in vivo data that I shared with you today, and Dr. Natalia Perez, who provided the cell-based data. And thank you all very much for your attention.
Thank you, Joanne. The slides will be available to everybody, so you can also see the conclusions that Joanne went very comprehensively over